Hi, everyone. Welcome to the International Science Festival in Gothenburg, which is one of Europe's leading popular science events and the only one of its kind in Sweden. My name is Miriam Frankel, and I'm the host this evening. I am science editor at The Conversation, which is a news and analysis publisher that is informed by research and aims to democratize knowledge. Now, tonight we celebrate the opening of the 25th International Science Festival, and that has been made possible thanks to a donation by Sten A. Olsson's Foundation for Research and Culture. So for 25 years, the festival has brought science to the public. It has sparked curiosity about research among adults as well as children and teenagers. So from today until Sunday, you are all welcome to join in our exciting digital program. And as always, this is free of charge and open to everyone. Now, the theme for this year's festival is Level Up. And that's the idea about the world's incompleteness and how we're always striving forward, really. But where exactly are we going? What is the next level, and how do we get there without harming the planet or the people who live here? So a scientist who has taken research to a whole new level is Professor Jennifer Doudna. She is a pioneer who stands for one of the greatest discoveries of modern science. She was awarded the Nobel Prize in Chemistry 2020, together with Emmanuelle Charpentier. And their Crispr technique has made it possible to essentially cut, paste, and edit in a genetic material, so DNA. It is a type of genetic scissors, therefore, that can be used to prevent genetic diseases or potentially cure certain types of cancer or HIV. But the ability to edit our genes has also given us the power to add new hereditary traits to humans. And that is an opportunity that leads to enormous and perhaps unmanageable ethical and philosophical challenges. So that's what we will be talking about tonight. You will be able to ask Jennifer questions during the last 20 minutes of this event. And you can do that by typing them into the little chat window to the right of the screen. But to kick off, and there's just so much to ask that I want to ask Jennifer. But just to kick off, Jennifer, would you mind just briefly describing what CRISPR is, how it works?
Well, it's wonderful to be here, Miriam. Thanks so much for hosting me in this incredible international celebration of science. It's awesome. And to kick things off, you mentioned CRISPR as a technology for editing DNA. And what I would point out first is that it's a technology that came from a fundamental question about biology, which is how do bacteria avoid viral infection? And it turns out one strategy they use is something called CRISPR that involves acquiring immunity to viruses that infect bacteria, and then using that acquired immunity to destroy virus genetic material, typically viral DNA. And it was through the process of discovering how that works from the molecular perspective, how do those molecular scissors actually function to cut DNA, that my collaborator Emmanuelle Charpentier and I and our students, our lab members, were able to understand that mechanism and harness it for a different purpose, namely for cutting DNA in animal or plant or human cells where it triggers DNA repair that can lead to changes to the genetic material in a precise fashion. In other words, triggering a change to a gene or a few genes in cells that allow scientists not only to discover genetic function, but importantly also to correct disease-causing mutations if we do this in the right kinds of human cells, for example.
Great. So, I mean, it is hard to sort of put into words just how momentous this invention really is. It is more or less given humans complete control of evolution. No longer do we have to wait or accept natural selection. We can wipe out entire species with a snip of this CRISPR scissors, or we can even potentially bring extinct species back to life. And of course, as we mentioned, to edit our own species to have the traits and features that we consider important. So at what point in developing this technology did it become clear to you just kind of what kind of power it could have?
Frankly, right away. I mean, right after we published our work in the summer of 2012, it was quickly apparent that the CRISPR technology would be adaptable to effectively any type of cell or organism. And that meant that in addition to allowing scientists to do new kinds of experiments, it also meant that it could be deployed to, as you implied, to alter DNA in human embryos, for example, where changes that occur in those cells become part of an entire individual if that embryo were to be implanted to create a pregnancy, and they can be passed on to future generations. So we call that a heritable change to the DNA. And this was done very early on in animals, for example, in mice. And there seemed to be no technical reason why it couldn't also be done in human beings, which is why I feel it's been critical over the last few years for the international scientific community to discuss this issue openly and figure out the right kinds of criteria that need to be in place for that type of work to go on in the future.
Yes, and that we're going to talk a lot more about that. But first, what kind of changes can we expect to be able to make with CRISPR sort of within the next decade or so? And perhaps more importantly, what kind of changes won't we be able to do? Because when we talk about CRISPR, we often imagine this world where not just will we be able to eradicate genetic diseases or any diseases, we could always also create so-called designer babies, babies with the right sort of height, eye color, intelligence that we're looking for. But for something sort of as hard to define as intelligence and many diseases too, there might be many genes involved, is that even something that is on the horizon anytime soon? So yeah, what's possible and what's not fairly soon?
Yeah, well, I think in the short term, we're already seeing uses of CRISPR to treat diseases that result from a single genetic mutation. And a great example of that is sickle cell disease, a well-known, well-documented genetic disease of humans that results from often a single base pair change, a single letter in the DNA of the human genome that when changed from what most people inherit, it gives rise to a form of the hemoglobin protein, the protein that carries our oxygen in red blood cells, that leads to those cells being sickled in shape and causes all kinds of physiological effects to affected people. Amazingly, the CRISPR technology is already in use in clinical trials to effectively cure this disease. In the cases so far, it's been used to activate fetal hemoglobin. So that effectively overrides the mutation that's present in affected patients. But in the future, it may also be possible to literally correct the disease-causing mutation, rewrite the DNA back to the type of sequence that most of us who don't have sickle cell disease will inherit. And I should point out that right now that's being done in individuals. It's not being done in embryos. It's being done in a way that's consistent with most types of therapies that affect an individual patient, but not their children. And I think that is a great example of the type of clinical application of CRISPR going forward, because it involves altering a single gene rather than many different genes. You alluded to, would it be possible, I don't know if it's desirable, but would it be possible to use CRISPR for more complex traits? And we could take a disease like schizophrenia, for example, well known to result from many different changes in the genome. And we don't, I think, fully understand how they all interact right now to give rise to a complex disease like schizophrenia. Would CRISPR be useful there? I think in the short term, primarily for research, for understanding the genetics of a disease like that, yet, but probably not in the near term as an actual therapy, because it will be difficult to deploy it to edit many genes at the same time in the affected cells, such as the brain. And then that gets to the more, sort of, I guess, philosophical question about, would one want to use this for altering traits that maybe don't affect our health directly, but might be considered desirable by some people? For example, could we alter people's height? Could we alter their intelligence? However that's very difficult to define, but however one might define that. And I think those are traits like intelligence clearly are highly complex. They result from many different genes. It's hard. I think any of us would probably be pressed even to define intelligence, and so CRISPR applied in that setting, I think, is not likely in the near future.
Okay, fair enough. So you talked a little bit about, changes made to embryos, sperm cells, and how many scientists are a bit uneasy about this. It comes with huge risks, and there's a lot we don't know about the genome as well, as you said. So these things can come with unintended consequences. So for that reason, a number of countries have actually banned gene editing of human embryos for use in assisted reproduction. Unfortunately, that doesn't necessarily stop individuals or companies to do this anyway. And in 2018, the first genome-edited babies were born in China, as you know, and the scientist behind it was eventually sentenced to prison. But I was wondering, what do we know about these babies today? And, without going too much into the ethics at this point, do you think that ultimately this accelerated or slowed down the research in the field?
Oh, I certainly think it slowed down research in terms of if we're talking about application of CRISPR in human embryos. I think it put a real damper on that, especially for folks that were perhaps considering doing it for clinical use. In other words, to create a pregnancy. That's clearly been, really received a very negative international reaction. And I think that has had, a dampening effect on the use of CRISPR in that type of setting, which is appropriate. I feel that needs to be treated with, extreme caution and care going forward. However, I think the good news is that there's a lot of interest in the use of CRISPR in that type of setting, and so research continues. And I think that responsible research into the use of CRISPR, even in human embryos, is appropriate if it's regulated accordingly. And that's what's happened really, I think, since 2018, is that there's a kind of a renewed international sense that as a community of scientists and clinicians, we need to work together to ensure that CRISPR is used responsibly in the future.
And do we know anything about these babies today? Have you been keeping up, finding out?
We don't, or at least I don't. It's very difficult because it's a difficult situation because, of course, they should be afforded appropriate privacy, I feel. And yet at the same time, one hopes that they are being monitored, that their health is being monitored appropriately, and that that they are informed of their health, the health implications of this procedure when they are at a point where they can take responsibility for their own health.
Okay. So moving on a bit to the ethics of the technology, in addition to the potentially, unintended consequences we talked about, there have been warnings, all sorts of warnings about, CRISPR having the potential to be used in biological warfare, potentially as an engineered virus, to also by tinkering with animals in the wild, it could wipe out entire ecosystems. There's also that slippery slope that we touched on between life-saving edits and, pure enhancement, and whether the technology could be used in unethical ways, such as eugenics. And so, given all of this, do you think it is immoral somehow to unleash this technology and go to the next level? Or is it perhaps more immoral to kind of fail to implement a technology that could help so many people who are suffering from horrific diseases? I know that your view has changed slightly on this over the years. And in your book, for example, you talk about how in a dream once Hitler asked you about CRISPR. So can you just talk us through your own kind of process and views on this?
Right. Well, as I mentioned earlier, the potential to use CRISPR in human embryos was clear from the those very early days of the technology. And I organized a meeting in early 2015 here in California that included about 20 scientists and clinicians to discuss this topic. And this was, before there was a lot of public attention to CRISPR, but many of us were, aware that this was a looming question in the field. Was, would and should this technology be utilized to create heritable changes in human beings? And what was one thing that stands out to me from that meeting is that there was, very, very active discussion around around kind of a big, conference table about this topic, with many people feeling, pointing out the challenges, the dangers. And then at one point, somebody leaned across the table and said, we might consider that at some point, we may feel that it's unethical not to use CRISPR to, prevent human suffering from known genetic diseases. And that just, everybody just kind of stopped at that point and said, whoa, yeah. This is like a way to just turn the tables on this whole question that we're discussing. And I think that's a really important point because, first of all, technology doesn't go backwards, right? We know this. You can't sort of unlearn or undiscover something that's been figured out. And nor I think would we want to, because I think that, the benefits of a technology like CRISPR vastly outweigh the risks, in my opinion, as long as, scientists are engaging and really, welcoming debate about this and involved in it. I don't think it's something we should throw over the fence to bioethicists and say, well, let somebody else worry about this. I think, all of us who've been involved in developing the technology in a way have a responsibility to be engaging in the discussion about it and certainly educating people about what it can and frankly can't do. So I think that's really, really key going forward.
So, but how have you gone from sort of worrying about it to maybe feeling a bit more comfortable with the potential of using this in some form? What's changed for you to kind of made you move a bit more towards potentially seeing the potential for some use for this?
Well, I certainly have always felt that the potential to use CRISPR in individuals is profoundly enabling, and I don't frankly see any ethical issues there beyond the way we would treat any type of therapy that somebody is given, whether it's a pill, a small molecule, or an antibody, or a genome editor, right? And because that affects the individual, and there are risks and benefits associated with that they have to decide about, but it doesn't affect anybody else directly. Whereas, using it in the human germline is a different story because that's creating a permanent change that, gets passed on. So I think that's an important distinction to make. And the vast majority of applications of CRISPR, certainly today, are for individuals. They're not in the germline. What's happening right now with germline work is primarily research, trying to understand even just the basic biology and genetics of early human development. So this is one thing that did start to change my views. I think early on for me, I was really resistant to the idea. I really felt that using CRISPR in human embryos, certainly for any kind of clinical application, but maybe even for certain research uses, it felt to me kind of wrong. And yet, as I learned from experts in the field that, first of all, there's very little information about early embryonic development because simply we didn't have tools to study it previously. I could see the argument, the value potentially for learning about that fundamental biology in a research setting using a technology like CRISPR under appropriate guidelines where embryos are treated with appropriate respect and limitations. I still feel that using it clinically, in other words, to implant edited embryos to create pregnancies, should not be done, certainly not right now. And there's a couple of important reasons for that. One is that the technology, I feel, itself is not ready. We still need to understand better how CRISPR functions in embryos, how DNA repair is dealt with in embryos. It is really not well understood. And a lot of that, frankly, can probably be learned from non-human embryos. And secondly, I think that there needs to be open, transparent societal discussion about this. Is this something that, we as a species want to be doing? Do we feel this is an appropriate way of applying the technology? And, the answer might be yes in some cases. And then we have to decide, how to proceed there. Is it going to be in the future the case that in vitro fertilization clinics, for example, might offer the CRISPR technology for certain types of editing? I think that's a scenario that could come to pass. And I think that people could possibly come to accept, much as just the IVF, the in vitro fertilization technique itself. I'm old enough, I remember, my parents sitting around the dinner table debating, test tube babies back when that was a brand new thing, right? And my parents, talking about how, they didn't think that was quite right. It really wasn't right that, embryos were being made in a test tube. That just seemed weird and wrong. And then what happened was, we had family friends who were unable to achieve a pregnancy on their own. And so they were assisted by a clinic like that. They had wonderful kids were normal and everybody was, great family. And my parents changed, as many people did, right? Yeah.
Indeed. I mean, do you think that something similar might happen? I mean, I was just wondering about the term designer babies. Is it a bit unfair? Kind of suggests that this is something entirely unnatural. And, after all, we already kind of design babies by choosing our mate. We take pregnancy supplements, we go to antenatal screenings, and we have been influencing the evolution of plants and animals since time immemorial. Look at the domestication of dogs. Dogs have genetic diseases because we have bred them to have the traits that we find desirable. So, and both both Sweden and the US have a history of forced sterilization of unwanted individuals, practice that was ultimately, luckily stopped. But so in a way, genetic design has already happened and it is happening. So do you think, the unease around CRISPR to some extent is just that kind of down to that irrational fear of technology? Or, how unnatural would you say it is compared to, as we're talking about, test tube babies, as they were called?
Right. Well, I think you make an important point that, human beings have been manipulating the genetics of organisms in our environment for, as long as there have been human beings, most likely, right? And certainly plant breeding goes back thousands of years. Somebody once showed me the precursor to a tomato plant, and believe me, it doesn't look anything like a modern tomato, and you probably wouldn't want to eat it. So, I think we would all agree that there are real benefits to being able to manipulate the genetics of organisms that and you pointed out the example of dogs, cats, other domesticated animals. I think what CRISPR does is it actually takes that to a new level, in the sense that, let's take plants, for example. So, traditionally plant breeders have to introduce some kind of, they have to figure out some way to introduce genetic change into seeds often. And then, and that's done randomly, right? And then you select for traits that are desirable. That process takes months to years. And of course, it brings along a lot of unknown, perhaps undesirable changes to the genetics. This is why we have thornless roses that don't have a nice smell anymore. We have tomatoes that last a long time on the store shelf, but they don't taste good anymore, things like that, right? And so imagine that with a tool like CRISPR, we now have a way to manipulate just those genes that will make desirable changes without changing anything else. And to do it a lot faster. Instead of it taking years and years to do this kind of manipulation, we can do it in a matter of weeks or months. And so I think for many people that are, kind of learning about this for the first time, when they really understand that, first of all, humans have been manipulating organisms for a long, long time, for eons, really. And also that CRISPR is a precision tool for doing this kind of manipulation. We still have to be, of course, thoughtful about how it's used. We have to be careful, but that's true also for, I would argue, for, more traditional ways of manipulating organisms in our environment.
Mm. Well, thanks very much for that. Another thing is the growing number of biohackers out there, and they're trying to edit their own genome using CRISPR, in the garage or whatever. For example, by injecting myostatin to try to grow muscles, all sorts of things. And many of them argue that, CRISPR should be less regulated, that everyone should be free to enhance themselves as they see fit, and that this would, ultimately make society more democratic. They worry that the alternative is a society, a market in which only certain individuals would have the means to edit their genes, and that would further boost the inequality we already have. What are your thoughts on, biohackers themselves, and also, this argument that, maybe is it every person's right?
Well, I certainly think that, making science more accessible to people is generally a good thing. I feel personally that, there's been a growing mistrust of science and scientists over the past few decades, really. And to try to reverse that trend, I'd like to see science integrated more into our, dinner time conversations, cocktail party discussions, and also to have technologies be more accessible, as long as they can be, conducted safely, for kids that are learning about science for the first time, because I think it makes science a lot more real for them. It makes it more exciting. It makes it more tangible. A great thing about CRISPR is that, as you implied, there are certain settings where it can be, I don't want to imply that it's easy to use, but it's relatively straightforward to use it. And so, for example, at the Innovative Genomics Institute that I started in the Bay Area, we've been working with high school teachers in our area to enable them to perform CRISPR-type editing experiments in bacteria or in yeast cells that these are experiments they can do with their students in the high school laboratories. I think this is a wonderful way to give students a hands-on feeling for a really exciting technology they might be reading about in the news, and they can now experience it and work with it themselves with their own hands. That's, in my opinion, that's great. Regarding biohackers and the scenario that you described of, people hacking their genes, et cetera, I think that's going to remain in the realm of science fiction, frankly, because, it's just not realistic to use CRISPR to alter our own genes right now in ways that would be meaningful. So I don't really see that happening. But the thing I like about the hacker movement is this kind of idea that, it's sort of science for all, right? Science shouldn't be sequestered away, siloed, and it's only practiced by experts who only talk to each other. I think it should be, socialized more than it has been recently.
Great. Okay, so I know you said that sort of designer babies and all this kind of editing intelligence and that sort of things is sort of far in the future. But if we just entertain the idea that sometime in the far future, everyone could edit their gene to make them taller, stronger, happier, more intelligent. It's interesting to think about, what such a society could be like. And I'm thinking in terms of evolution, would that not reduce genetic diversity? You know, the reason evolution has been so successful is that it produces all this diversity. And suddenly, if there's an ecological disaster, some individuals are wiped out, others thrive. And it's kind of helped our species survive. So from an existential perspective, you know, if we all became more homogenous, albeit smarter, you know, is there an inherent risk that we could be wiped out more easily as a species?
Well, honestly, I'm much more concerned about things like climate change that, you know, are truly existential risks that, you know, we're facing right now. I think the scenario you describe is again, it's really fiction, because, you know, if you try to imagine how would that actually come to pass? I mean, you know, a great science fiction writer could write a wonderful story about that happening, you know, many years in the future. But it's going to take, even if we all said, this is a great idea, let's all start editing our embryos today, it would take a really, really long time to do what you're describing. However, I do want to point out for folks that are listening to this and, you know, thinking about the genetics of organisms that, you know, you do describe an important, you know, aspect of biology that we do need to think about. I don't think it's important to think about that for humans right now, because it's just not really a possibility, but for other organisms. And, you know, lots of examples of this, like let's think about bananas. You know, bananas have been threatened in our environment by pathogens, you know, by fungi and other types of diseases that affect them because their genetics are very, very similar, basically identical in all bananas that are grown commercially right now. And so that's a real problem. And so again, I think CRISPR can help in a way, right? Not hurt, because I think it can help to actually introduce more genetic diversity into certain kinds of organisms that will give them resistance to disease. And, you know, we think about this right now a lot in the context of climate change, because with the climate changing rapidly, organisms are having to adapt faster than potentially they could do naturally by evolution, right? And so to avoid, you know, extinction of certain species, it actually may be beneficial to help them along in a way with CRISPR.
Well, that's a fair point. But I'm just not going to give up on this science fiction society just yet. So, you know, again, let's just think about, you know, if everyone is tall and smart and so forth, it sort of removes the advantages you'd think of being tall or smart, if that's something that you value. So, you know, how would it work in terms of, you know, would we get people to do all the jobs and chores necessary if everyone was sort of overqualified? Also, I was wondering what your thoughts are on kind of people being different and outsiders from a sort of psychological view. You know, if everyone sort of fits in, if everyone has the right traits, you know, a lot of human creativity, ingenuity comes from people who've sort of felt outside. I think outsiders. I think you've even spoken about this sometimes, you know. And so, you know, human success isn't just down to muscles and brains, you know, it's down to a lot of different things like creativity, grit, resilience, you know. How do you see CRISPR fit in with those things?
Well, again, I think, you know, like many people, I think we value the interesting diversity that we see across humanity. It's really something that I think as human beings, we intrinsically think is interesting about our species, is that we're not all identical. We're not all the same. We're not all clones of each other. And I think that's a wonderful aspect of life that, you know, we want to embrace rather than run away from or try to change, in my view. That being said, I think that we can certainly imagine that there might be certain types of afflictions that would be potentially good to eliminate from the human population. And, you know, we think about diseases like, well, sickle cell is, of course, one that we talked about earlier, but what about cystic fibrosis? What about muscular dystrophy? What about Huntington's disease? What about Alzheimer's disease? What about heart disease? You know, these are all things that, you know, if there were a way to genetically protect the human population from diseases of that type, I think many folks would at least think about it, right? They would at least consider, is that something that maybe we do want to do? We don't want to affect the rest of the diversity that we see across humanity, but we want to find ways to avoid human suffering.
Mm. Yeah, it's a fine balance there, isn't it? So there are also, there are so many kinds of potential uses for gene editing. We've only touched on very few, everything from agriculture, potentially help us fight pandemics like COVID or prevent, or, to grow sort of human organs in animals, With all of those, what do you think are sort of the top most exciting ones, and which ones are you the most worried about?
Well, I'm certainly very excited about the potential to cure monogenetic diseases, diseases that result from a single well-known, well-defined mutation where there's good evidence that by making a change to that gene, someone can be restored to health. So I think that's the kind of use of CRISPR that in the coming years is, first of all, the most accessible, and secondly, will have the greatest impact if, and there's an important if here, and that is if those applications of CRISPR are affordable and accessible to people, right? Let's assume they're going to be safe and effective, and of course that would have to be tested and demonstrated. But beyond that, if those treatments still cost, $2 million US, for per patient, they will just not be widely impactful because, many people that need those treatments globally won't be able to afford them or to get access to them. So that's something I think about daily, really. I think about, how do we go from where we are today? We've got an exciting technology in the hands of scientists, many researchers benefiting from, the kind of work they can now do with the technology, and some, beginning clinical uses of the technology. But how in the future, how do we make sure that this becomes, for example, a standard of care? What if it someday is a standard of care that somebody who is, say, susceptible to Alzheimer's disease has an opportunity to use genome editing to alter their, just a couple of genes that might be protective against Alzheimer's. That would be extraordinary if that could actually happen. And it still feels to me like that's a ways away, but we have to work towards that vision, I think.
So is there anything, though, that really gives you nightmares? Any use that you would just right now say, this is something that we definitely should stay away from?
Well, I think we have to be very cautious about environmental uses of CRISPR. We didn't talk about this yet, but one of the applications of CRISPR that's attracted attention and could be beneficial, but also comes with risk, is using it in a format that's been termed a gene drive, where you give to an organism a gene, or you use CRISPR to create some kind of a genetic change, and at the same time, you give the organism the CRISPR technology, molecules themselves, so that those can go off and create that same change in another organism. And so, this has been applied in organisms like fruit flies, in mosquitoes. In mosquitoes, there's the vision that a gene drive could be useful for creating sterile male mosquitoes, for example, that can't propagate. So what if you could eliminate species of mosquitoes that are responsible for devastating diseases in humans, like malaria? This could have an enormous beneficial, human health, public health kind of impact globally if it could be done. On the flip side, there's the risk that goes along with something like that. Could it get out of control? Do we really know what the impact would be of eliminating a species, for example? And so I think that's it's not that I feel that should never be done. It's just that I think it has to be, approached with suitable caution and appropriate regulation.
Okay, so just lastly, before we open up to questions, the regulations surrounding gene editing, as we've touched on, is patchy and often ambiguous. Are you in favor? What should we do? Are you in favor of something like a global moratorium? I take it not, maybe? I mean, what do you think would be the best way to?
Oh, no, I certainly don't favor a global moratorium. I think the word moratorium implies enforcement, for one thing, and I don't really know how one would, even if that were a desirable thing, I don't know how you would enforce it. But I don't feel it's desirable. I don't. I think science and technology is, it's beneficial ultimately for the human species. That's been shown over and over. That new technologies are the engine that drive new discoveries, and then vice versa. New discoveries will, fuel technologies of the future. And so I don't think we want to go backwards or run away from that. But I do think that we want to be quite actively engaging in the development of technology. And I'll give you a parallel right now. So a lot of discussion, as you know, about artificial intelligence, about machine learning, where's that headed? And I think there's some interesting parallels to CRISPR in that regard, because again, there's a movement among at least some computer scientists to be actively engaging in the public conversation around artificial intelligence. How do we, and I think many people feel that there's a lot of benefit to come from the developments there, but also that we need to be cautious and we need to be responsible in the way that work develops.
Excellent. Thank you so much for that. We are now opening up to questions. Have we got any questions coming in? I have not received any so far. I could maybe just ask you then, for now, what you and Emmanuelle were two women for the first time sharing the chemistry Nobel Prize. What do you think about the sort of leaky pipelines, as is often described, of women in science, where you get a lot of women at sort of university student level, and then they sort of tend to disappear as you go later in the career. And, just the lack of women role models. How has your own experience been as a woman in science, and how do you feel about this topic?
Well, I think it's important to create a culture in science that is more inclusive and more kind of accepting of different ways of doing science. I think if we do that effectively, we will see a lot more women and just a lot more diverse, types of scientists staying in the field, just because the field will then be enabling to them. I think in the case of women, you pointed out the leaky pipeline. This has been commented on. I mean, I remember, this was discussed back when I was a student, a few years ago. So, it's not a problem that's new. And there's been lots of debate. There's probably many reasons for it. But I do think that, in my own experience, I think for many women are when they get to a point in their career where they might become an independent faculty member, an independent scientist, leading a group of researchers, that often coincides with their childbearing years, with needs of elderly or aging parents, where they are often the lead caregiver or have to organize that care. That was certainly true for me, both of those things. And, it certainly really helps to have a supportive partner, which I do. I'm very lucky. I have a wonderful husband who's also a scientist and who's always been, super supportive of my work and my career. But I think it also is very helpful if you have colleagues and, kind of a work environment that is flexible enough to accommodate needs like that. And that's, it also is true for men, of course, but I think traditionally, at least, it's been the women that are more affected by those kinds of, life events that just, can have an impact on their professional advancement.
Okay. Thank you for that. I have now got a number of questions, so I'll get cracking. We've got Daniel, who has asked, I really want to know, he says, how did you feel when you got the message that you and Emmanuelle had won the Nobel Prize?
Total shock. I was awakened from a deep sleep, and you can probably imagine maybe the feeling of, waking up, and it's the wee hours here in California, and a voice on the phone is giving me this news. And I actually, I kind of didn't believe it initially. And but right away, the very first other call that I got was from Martin Yinek, who is the scientist in my lab who did the research on CRISPR-Cas9 in our collaboration with Emmanuelle. He's now a faculty member in Zurich. And so when I saw that he was calling me from Zurich at 3:00 in the morning in California, I knew it was real.
Oh God, that must be. So I remember as a journalist, kind of the year before, or the 2 years before, it was talked about that the chemistry one might be CRISPR, and then finally it was. We've also got Arian, who has asked, how much can we reduce the off-target effects of genome editing? So the consequence,
right.
yeah.
Yeah, it's a great question, because as you can imagine, CRISPR isn't perfect, and it could, in principle, and we know in practice, it can cleave DNA or trigger genome edits at unintended positions in the DNA. And so what do we do about that? And there's been a couple of interesting, well, I'll just mention a couple of things. One is that, first of all, there's been a lot of research on that topic. So we understand now a lot about the different factors that affect the accuracy of CRISPR. And one obvious one is simply choosing guide RNA, which are the molecules that direct the CRISPR-Cas9 protein to a particular DNA sequence for where it does whatever it does, whether it's cutting or doing something else. Selecting those guide RNAs to purposely avoid closely matching sequences elsewhere in the genome. So that's, one strategy that, has been effective. And then I think that the other thing that's happened is that there's been a lot of work to understand, not only to understand how the natural CRISPR enzymes work, but also to engineer new forms that will have higher levels of accuracy. And frankly, they often just simply have lower levels of overall activity, which then of course means that there are fewer, undesired off-target events as well. And that's also been an effective way to avoid off-target. So I currently don't feel that is a bottleneck for CRISPR. I think it's something we have to pay attention to. We have to be able to monitor it, especially for clinical use. But I don't see it as a gating aspect of CRISPR.
Great. We've also got Sarah asking, can you add a missing gene only to an embryo or to an older child and still make a difference? Could you add a missing gene? Yes. I mean, in principle, the answer is yes with CRISPR, right? And you could potentially do that either in an embryo or in an older child where it would affect just that individual. Now, you're touching on one of the, I think, currently the sort of areas of active development with CRISPR, which is that it's relatively straightforward to disrupt a gene. It's more difficult to insert a new gene. It's possible, but it's harder to do that in the context of a clinical setting where you'd like to have every cell that needs that new gene to get it, right? And so just ensuring that kind of efficiency and also the targeting of particular cell types in an individual, very difficult to do right now. So the way that's being done currently is to do it in cells that can be extracted from a patient, like sickle cell disease, back to that example. The cells can be edited in the laboratory, and they can be either, a gene can be knocked out, or a gene can be activated, or a new gene or a corrected gene can be inserted. So all of those things are possible in the lab, where you can monitor the editing and make sure it's done, it's accurate. And then those edited cells can be transplanted back into the patient. And I'm hoping that, that's the current, kind of one of the reasons why those types of blood disorders are being treated using CRISPR currently. But I think in the future, hopefully the not too distant future, we will have delivery
vehicles for CRISPR, for genome editors, that will allow the kind of editing that will enable correction of other kinds of genetic mutations, such as, for cystic fibrosis, et cetera. But we'd have to have delivery vehicles that allow correction in lung cells or in muscle cells or in the brain.
Okay. And how far away are we from these?
Well, some of them are, in some cases that maybe not too far. I mean, the case of muscular dystrophy, I think there's been enormous progress made there, both in academic labs and in companies, because of the ability to use certain kinds of viruses that can deliver CRISPR molecules into muscle cells, because that's the kind of cell that they normally infect. And so this is a strategy that, has been already deployed in animals effectively, and now I think is at least getting to the point where one could consider doing human trials with that kind of approach.
Exciting. We've also got Heba asking, what advice do you give to early career researchers, especially if they live in less developed countries with fewer opportunities in research? It's a good question.
It's a great question. Yeah, I myself grew up in the U.S., but I grew up in Hawaii, which is, I often felt like I was in a little rock in the middle of a big ocean, far away from all the action, which is kind of true. It was certainly a beautiful place to grow up, but there weren't any scientists in my family, and so I had to kind of make my own way. I had to follow my own interests, and I had to find people who would support my interests. I was fortunate in a way because my father was a faculty member at the University of Hawaii, and even though he was not a scientist, he was a literature professor, but he saw that I was really interested in science and chemistry and biology. And so he introduced me to colleagues of his that gave me some opportunities to work in their labs, when I was still in high school. I think that, wherever you are in the world and whatever your circumstances, if you are truly passionate about something and you truly, reach out and look for opportunities to, advance your interests and find mentors who will support you, I think you can make progress. I know that's kind of an American way of looking at the world, but I feel it's true because I have known people in various parts of the world in quite some of them in quite limited circumstances who nonetheless were able to make progress and follow their passions, and in some cases make really big impacts in their fields because they were very driven and focused on what they wanted to do.
Yeah, I mean, I don't think I've ever met a scientist who wasn't really passionate about it, or a successful one anyway. It's definitely is a prerequisite, isn't it? What else have we got? We have got a question about several people have asked. We did touch on it a bit, but maybe you would like to expand a bit about how CRISPR, or will CRISPR affect evolution or natural selection ultimately?
Well, I think it certainly will affect evolution and natural selection of some organisms, in our environment. Plants, I think likely, some kinds of animals, I think that's likely. And, but you may be asking about humans. Do I think that will happen? And as I said during our discussion earlier, I think that's not happening anytime soon, certainly not in my lifetime, and probably not for quite a while afterwards as well, because it's just, and again, even if we said that's a great idea, we're going to do that, we're going to engineer humans, we're going to start today, we're going to engineer every single person on the planet, think about how long that would take. It would take generations really for that to start to have a real impact on human beings. Now, that being said, I still think it's critical that we be, thinking about the use of CRISPR and figuring out how to be responsible in its use in heritable, human genome editing, because it will happen, right? There's no doubt, in my mind, at least. I think this will happen. There will be companies, there will be IVF clinics that are going to offer this. Mm-hmm. And that doesn't mean that I think that immediately the whole human population will be, edited and that it'll affect human evolution immediately. But it does have real, I think, very real, kind of profound implications for things like equity, who gets access, who decides, who pays for it. Do some people get to do this and other people don't? And then what would be the impact of that? I think those are all, very real possibilities that must be considered and discussed.
Okay, we might have time for one more question, then we'll have to wrap up. So I think this would be a good one to end on. What is your lab working on at the moment? What are you looking to achieve next?
Right. Well, two things in my own lab, and then some bigger projects that we're working on in the context of our institute and our university. So in my own lab, two things. One is that we're continuing to understand and discover the fundamentals of CRISPR biology. I think there's a lot still to be learned about the way these systems operate naturally in microbes. We work a lot with Jillian Banfield at Berkeley, a professor of geobiology. So she researches lots of microbes that that grow in the environment and tries to understand their biology. Many of them have CRISPR systems. We've recently found that a lot of bacteriophage have CRISPR systems. And so I think that's an area where there's still a lot to be discovered. And so we're working in that on that part of the field. And then we're also very actively working on this whole issue of delivery. How do we deliver genome editing molecules into certain types of cells, whether it's a plant cell or an animal cell or a human tissue type? Those are all aspects of CRISPR that will be greatly enabled with better ways of delivering CRISPR. So that's the other thing that we're doing. And then in a broader sense, I'll just mention that at the Innovative Genomics Institute, we're focused on both affordable, accessible uses of CRISPR for clinical applications, as well as looking at opportunities to use CRISPR to mitigate the effects of climate change, both areas where I think there's potentially huge impact if we can figure out how to deploy CRISPR in ways that are beneficial and again are accessible to people around the globe.
Excellent. That is super interesting. I mean, sometimes when we talk about this, we talk about, the end of evolution, the wide existential risk, but actually it could also help us save the planet, as you talked about there. Jennifer, thank you so much for talking to us today. I mean, I know that you are really passionate about going out and talking to people about this. You want people to continue discussing it, having an opinion. So I really hope that everyone who has been listening has, been able to take something away from this. And when you have cocktails later, that you can continue to discuss this. So I think that we will wrap up there. Thanks to everyone who came to listen, and thanks most of all to you, Jennifer. And I hope you all have a lovely evening.
Thank you, Miriam, and thanks to everybody who plugged in to listen.